All right, welcome to part two for the exercise metabolism talk. We're going to pick up right where we left off last time, which was after phase three of the complete breakdown of a carbohydrate molecule or a glucose molecule. So if you recall, we were going from pyruvic acid or pyruvate to acetyl-CoA. Now we're at stage three, which is the magic of the Krebs cycle. Uh, Krebs cycle has multiple names: tricarboxylic acid cycle, citric acid cycle. Krebs cycle seems to have stuck um, for the most part, so we'll use that term. Uh, the Krebs cycle begins with oxaloacetate. It is a cycle, so it kind of begins and ends with that if you want to look at it in that way. It's an 8-step process in the mitochondrial matrix. So one thing to be mindful of, now we are in the mitochondria, and so the rest of the processes that occur metabolically from this point forward are going on in the mitochondria, unless I say otherwise. This process doesn't, so the Krebs cycle doesn't use ATP. It only produces one ATP, and a lot of students sort of react to that. It's like, oh, I thought the Krebs cycle was this massive producer of ATP. Not so much. One turn of the Krebs cycle, and we get 2 turns per glucose molecule because we ended up with 2-acetyl-CoA, only gives you 2 ATP. But maybe more importantly, what the Krebs cycle does provide us with for each acetyl-CoA that enters is 3 NADH and 1 FADH, which do give us plenty of ATP in our final step. We do get 2 carbon dioxides produced for each turn, and we still are not using oxygen. However, the process of the Krebs cycle would not occur unless there was oxygen present in the mitochondria. So here's a pretty standard, with detail, picture of the reactions and the substrates involved in the Krebs cycle. There's a lot. I'm not going to cover all of them, and all I'm really going to sort of at least highlight is, yeah, okay, we start off with oxaloacetate over here. We have the addition or the contribution of acetyl-CoA that sort of continues this cycle to keep spinning, or this cycle to keep spinning, I guess is a better term. Some important sort of byproducts and where they occur right here in the isocitrate reaction. We do have the production of an NADH and also the elimination of a carbon dioxide. I guess it's a production, but then elimination of carbon dioxide. And then the alpha ketoglutarate reaction, or alpha ketoglutarate dehydrogenase reaction, gives us our second NADH and our second carbon dioxide. And so if you've been playing along, some people like to count. Our glucose started with 6 carbon. By all accounts, acetyl-CoA was a 2-carbon structure. Now we've gotten rid of 2 carbons. Our carbohydrate, our glucose molecule that was once 6 carbons joined together, is now done. It's pretty much gone, and there's no remnants of it, so to speak, left over. Those last 2 carbons were the final holdouts. As we continue on through the cycle, we do have the production of ATP. As we make our way back up, we have pretty much our only presence of FADH in this process. And so we do have the production of an FADH molecule and then our final NADH molecule. That's all we're going to really talk about now in terms of the mechanics of the Krebs cycle. We'll get back to it a little bit with our discussion on carbohydrates, but that won't be for a few minutes. So summary of the Krebs cycle, yeah, it's pretty much straight up what we just talked about. For each turn, and be mindful that the Krebs cycle will turn twice because we end up with 2-acetyl-CoA per glucose. We end up with 3 NADH, 1 FADH, 1 ATP, and 2 carbon dioxide. Wow, such quick progress. We are now at our final stage, stage 4 of the complete metabolism of a carbohydrate molecule, of a glucose molecule, and that would be the electron transport chain. So up until this point, everything has been setting the stage for this massive onslaught of ATP release or reformation, however you want to state it. We're going to get it. So the electron transport chain, often referred to as oxidative phosphorylation, yes, they're synonymous, is the final metabolic pathway. It's happening in the inner membrane of the cristae of the mitochondria. ATP is formed from ADP and inorganic phosphate, just like it kind of has been throughout, right? It's just bringing that phosphate back to its sort of home molecule, so to speak. Hydrogen ions and electrons come from previous stages, and that is in the form of NADH and FADH, right? So I keep mentioning them, right? And I keep being careful to always remember when to talk about them when they're formed. This is why, because for each NADH molecule that makes its way to the electron transport chain, we get 3 ATP. For each FADH, we get 2. The final electron acceptor of this chain is oxygen, and from it we get water. And so if I ever ask you what the end product of the complete breakdown of a carbohydrate molecule is, it's going to be water. Carbon dioxide's in there as well, but really water is the end, very, very end product. Yeah, so the drawn out version, and again, there's some detail here, but man, there could be so much more detail. Starting from the upper left, and the electron transport chain is really this almost a magnetic process of pulling these NADHs and FADHs across membranes. And when they're pulled across, we have this phosphorylation of ADP to get the phosphate back on it. And so if you start up here in the upper left, we have our NADH molecule. It will donate some electrons and ultimately end up with ATP from ADP and PI, and that ends up converting NADH to FADH. From FADH, we go through a whole bunch of smaller reactions and ultimately get to a point where it gets pulled across a membrane again. We get another ATP. All that happens again, gets pulled across another membrane, and we get a third ATP. So if we started with NADH, we do end up with 3 ATP. If we start with FADH, we kind of skip the first step, right? So we only end up with 2 ATP per FADH. So that's kind of the explanation. It just starts later down the chain, and therefore it doesn't get as many ATP as NADH, which starts farther up. So finally, and maybe I should mention, I'll go back, the magic of the computer, right? At the very, very end here, we have our oxygen molecule, and it's a half of an oxygen molecule, so you can just kind of get rid of the 2. And we have it combining with hydrogen, and we end up with the formation of water. That's it. We've completed our 4-stage journey. So if we were to tally the ATP generated from the catabolism of a molecule of glucose, the total would be 38. Usually I would spend some time talking about each one of these, but just freeze the screen and look at these numbers. Glycolysis, and so we have the high energy products formed in each of the stages. We have the ATP from oxidative phosphorylation, so what we're getting from the NADH and the FADH, and then we have the subtotal of ATP. Put them all together, the numbers, they add up. 38 is going to be the total from glycolysis. This assumes we're using the malate aspartate shuttle and not the glycerol phosphate shuttle, which is kind of a big assumption. This is also assuming that we're using glucose, we're calling it glycolysis, glucose instead of glycogen as a starting point, right? So if we use glycogen, it'd be one more, it'd be 39. Why is that? Because that phosphorylase reaction instead of the hexokinase reaction. So know those little details, but that 38 number is something that you should be able to calculate, right? Let's move on over to the left. So we keep coming back to this old school graphic because it's just so useful, right? So let's talk about the catabolism of fat, right? When we talk about exercise, which we'll talk a little bit more about exercise specifically later in this talk, but it's usually carbohydrates and fats that we talk about the breakdown of. We don't talk about proteins much. Fats can contribute significantly to exercise metabolism. Storage form of fat is triglycerides. You should know that from the digestion discussion. And triglycerides are broken down when they start to be enzymatically broken down into fatty acids and glycerol. Glycerol is pretty cool because it goes back over and joins the second half of glycolysis. So we pretty much get half of a sugar molecule from the glycerol backbone of a triglyceride. And glycerol is a 3-carbon molecule, and off of each one of those carbons is this free fatty acid chain. So we haven't talked much about fats yet. We have a whole chapter dedicated to them later, but at least for now, know that glycerol is a 3-carbon structure. It's literally half of a glucose, and that's why it's so easy for it to enter the second phase of glycolysis, because that's after that 6-carbon glucose molecule has been split into 2. So the fatty acids go through a process called beta oxidation. It gives us some magic, but one of the byproducts of beta oxidation, we'll talk about the magic in a second. One of the byproducts of beta oxidation is the formation of acetyl-CoA, our wonderful universal intermediate of metabolism, and that will, of course, make its way down the Krebs cycle and to the electron transport chain. So let's talk lipolysis. Lipolysis is a term that describes the breakdown of, or the catabolism of, fats. So the breakdown of triacylglycerol, I think for the most part they're still being called triglycerides. People in the science world just like to change names around a bit. They're both the same molecule, so depending. Triglyceride just makes sense, but to me, I'm old school, I guess. So the breakdown of triglycerides to free fatty acids and glycerol. And so where are we finding these triglyceride molecules? Well, we know we store them in muscle cells. They're circulating in our blood, and they're also found in our adipose tissue. So we have our fat tissue, our muscle tissue, and we have them in our fat tissues. And from our fat tissues, they do get liberated and spill into, or deposited into, our bloodstream, and are then delivered to our muscles to be used as a fuel source then. The free fatty acids, FFAs, are catabolized in the mitochondria, and that four-reaction pathway that breaks them down is called beta oxidation, and we'll talk about that in the next couple slides. Firstly, let's talk about releasing the glycerol from the fatty acids, the enzyme lipoprotein lipase. And I don't know enough about its dual role and its role in basically doing the exact opposite thing. Lipoprotein lipase does play an important role in the storage of body fat. We know it works in conjunction with insulin. We know it helps us store body fat. However, the same enzyme also helps with breaking the free fatty acids off of glycerol with the help of water. So we basically have a triglyceride molecule, glycerol backbone, remember, 3-carbon structure with these long fatty acids hanging off of it. With the help of water and this enzyme lipoprotein lipase, the glycerol gets basically torn off, or maybe it's the opposite, the free fatty acids get torn off the glycerol, and we end up with pretty much 4 molecules, a glycerol and 3 free fatty acids. Lipoprotein lipase is an allosteric enzyme, so it can be turned on and off. Hormone-sensitive lipase plays a huge role in activating lipoprotein lipase in its role to break down fats and liberate fats. Things like epinephrine, norepinephrine, glucagon, human growth hormone all do the same thing, but really that hormone-sensitive lipase is a pretty key player. So beta oxidation. So once we've separated our glycerol, I already told you, yeah, glycerol goes off into the glycolysis, the second half. Now we have these free fatty acids. What do we do with these massive chains of carbons with a whole bunch of hydrogens attached to them? Well, beta oxidation is what takes care of it. We're not going to talk about all the reactions of beta oxidation, but we're at least going to highlight what's the purpose of beta oxidation. And if you look at this free fatty acid at the bottom of the screen, what number of carbons would you think would be most beneficial for our metabolism to use right away? Think about it for a second. Do we want to make it a 6-carbon? That could give us a glucose, right? Do we want to go 3? That's like half a glucose. It's almost like making another glycerol. Or do we want to go 2-carbon? What was that 2-carbon structure that we've talked about quite a bit? Oh, yes, acetyl-CoA. That is what beta oxidation aims to make, is a lot of acetyl-CoA. So the process of beta oxidation cuts off 2-carbon fragments at a time, just chops them. So we end up with these 2-carbon molecules floating around, and we'll move forward to the next screen, I guess. And those 2-carbon molecules become acetyl-CoA ultimately. And as you already know, the whole role of acetyl-CoA through the Krebs cycle and electron transport chain, full-on ATP powerhouse. So looking at this graphic, if we start up in the upper left, yeah, we have glucose and glycolysis and all that stuff. The only reason that's really here for this purpose is, okay, let's look at our triglyceride molecule. That's what we're after, right? We're talking about breakdown of fats. So that triglyceride molecule plus 3 water and lipoprotein lipase give us that glycerol, and the glycerol goes back to 3-phosphoglyceraldehyde, which is the beginning of that second phase of glycolysis, right? When that 6-carbon glucose structure got split into 2 3-carbon ones. That's where glycerol goes. And so we get the whole 19 ATP from that. And then we end up with our 3 fatty acids, right? And there's that picture of the fatty acid on the previous screen. Those 3 fatty acids go through this process of beta oxidation, and it just spins, right? It's almost like a saw or like a cleaver, just chopping off 2 at a time. Just keep chopping until that fatty acid is down to no extra fragments to break off. I believe all free fatty acids are in even numbers. Don't quote me on that though. So there's no issue with a single carbon floating around by itself. Um, the byproducts of beta oxidation, yes, that acetyl molecule which combines with CoA to give us acetyl-CoA, that's huge. Um, we do get some hydrogen transported to the electron transport chain. As you could expect, those are transported via the NADH. So we do form NADH in beta oxidation as well. Oh, that's like bonus ATP, right? This little table here at the bottom is kind of cool to look at, just to give us an idea of how much ATP we get from an 18 carbon free triglyceride, so free fatty acid molecule. And if we look at the total number of ATP on the far right here, from one molecule of glycerol, yes, we get 19 ATP. You should have been able to calculate that from the glycolysis one, right? That we just did, the whole 38. It's half of 38. But then from the 3 molecules of the 18 carbon fatty acids, beta oxidation and the Krebs cycle, electron transport chain, we end up with 441. Add the two together, we get 460 ATP from one fat molecule. And that's actually a relatively short one. There's longer fat molecules, which could add another 50, 60, 70. So one molecule of glucose, 38 ATP, one molecule of fat, 460. It's an incredibly efficient way for our body to store energy. At least that's the take-home message. Getting to that energy, getting to this process of beta oxidation is a little bit trickier. It is not as easily accessed as glycolysis. It obviously requires oxygen to be present in the mitochondria. It requires a role of carnitine, and carnitine plays an important role of basically getting those fats to the mitochondria. So it's not that simply done. It's a carnitine shuttle. People supplement carnitine. It's an amino acid, right? To see if that might be a limiting factor in getting fats into the cells. And supplementation doesn't do that much. It's okay. I don't want to totally debunk it, but so the access to fat is not nearly as easy as it is to glucose in terms of metabolism. So when we're in resting conditions, plenty of oxygen in the cell, no need to rush with that ATP production, fats are easy to get to. As soon as we start doing meaningful exercise and we need ATP at a fast rate, guess what? Fats kind of fall behind. The rate of ATP production from the breakdown of fats is slower compared to carbohydrates. And so when we need ATP fast, when we're stepping on that gas pedal and we need a lot of energy, fats can't keep up. And so there ends up being a mix, carbohydrate and fat breakdown. Let's talk a little bit about protein first before we get into more sort of exercise related stuff. So proteins, yeah, we have quite a bit stored. If you recall that table we had, seems like eons ago, right, where it compared how much protein we have in our body and how long we could exercise using it. It's quite a while. Not nearly as long as fats, but way more protein stored than we do sugars, right, and carbohydrates. Proteins, thousands of different protein molecules. Ultimately, they're all broken down into their basic amino acid structures, which we have 20 amino acids. And in that process, they can contribute in multiple ways to metabolism. So yeah, you'll see there is 3 arrows. They can go and actually become pyruvate. Yeah. They can become acetyl-CoA, and they can go straight to helping out with the Krebs cycle. So a lot to talk about in terms of understanding the role of amino acids in metabolism. Yes, a very wordy slide. During catabolism, amino acids can be used to form other molecules or used to form other amino acids. And so the process of deamination is the removal of an amine group from an amino acid. Don't worry too much about this now. When we talk about amino acids, we'll talk about it a lot. And then when that is moved to another amino acid or to form a new amino acid, we call that transamination. So our body can make amino acids. The non-essential amino acids, there's 11 of them, our body can make those quite easily. The essential ones, not so much when we have to consume them in our diet. Again, we'll talk about all about the food aspect of amino acids a little bit later in the semester. When an amino acid is deaminated, so when the amine group is taken off of the amino acid, the carbon chain can actually be used to form a molecule of glycolysis or go into the tricarboxylic acid cycle, which is the Krebs cycle, and ATP is generated from that. And we call that amino acid oxidation. So basically the oxidation of amino acids, so the use of oxidative metabolism to break down an amino acid and get ATP from it. The term gluconeogenesis is used to describe the process of making amino acids, or to make a process of making glucose from amino acids, and that's a pretty big deal. And individuals who train for long durations and high intensities, or both, and both, individuals who are in a low carbohydrate nutritional status, people who are going ketogenic, right? Ketogenic diets are everything right now. Everyone's keto. Even like athletes are going keto. Why? Guess what happens? Our body goes, well, you know what? If you're not going to feed me carbohydrates while I'm training and when I need them the most, or if you're just not going to feed them to me in general, but even if I'm not training, why don't we tap into something else to make carbohydrates from? And that would be amino acids. So it's very, very difficult to be in a low carbohydrate status situation, whether it's through training or through diet or both, and maintaining muscle mass. Because guess what your muscles become? Your body's favorite fuel source. And so it's a big deal. We'll talk about it throughout the semester, but at least be mindful the process of gluconeogenesis happens and it's accelerated in those conditions through extreme training or extreme carbohydrate depletion. How much does amino acid oxidation contribute to exercise metabolism? There's a fair amount of research out there on this, and values range with confidence. Moderate intensity, endurance exercise, probably in the neighborhood of 5% of overall energy. As exercise intensity increases and as exercise duration increases, that can be anywhere from 5% up to 10%. So we're looking at a fair amount of energy coming from amino acids. If you take some, ultra distance type people and they're doing 3-4 hours of training at a pretty good clip, even if they're supplementing carbohydrates during that training, it's probably not enough. And so they're going to take a bite out of their muscles, so to speak, to help contribute to that carbohydrate as a fuel source. So lots of gluconeogenesis going on. Yeah, here's a graphic. This is the Krebs cycle again, and it's probably the most abbreviated form you'll ever see, but that's a Krebs cycle down there in the middle of that cycle. In the colored boxes, the red and the yellow, we have the contributions of different amino acids in the process of the Krebs cycle. And actually up here we do have pyruvate, so coming from glycolysis. So the contribution of amino acids in that process of metabolism. So this is a situation where amino acids are contributing to forming the substrates needed for the functioning of the reactions of this process. So it's not necessarily the oxidation of amino acids. So this isn't saying, oh, these amino acids are being converted to ATP. More that these amino acids are necessary for the reactions to break down fats, to break down carbohydrates to occur, right? So they kind of play maybe a backseat role. It's like, oh yeah, we take for granted that all these substrates exist to help us with the 9 steps of glycolysis and the 8 steps of the Krebs cycle. Who are those players? They're pretty much amino acids. And so it gives, hopefully this gives you a better understanding. It's like, oh yeah, even though I'm not necessarily breaking proteins down specifically for energy, and let's say I'm pretty well carbohydrate fed, I don't need to use proteins as a fuel source if I'm consuming carbohydrates, but for sure I need to use proteins to help the process of breaking down carbohydrates and fats. So there's a lot of them on this list. The branch chain amino acids typically stand out: isoleucine, leucine, valine. They're kind of all over the place in this, right? The branch chain amino acids lately are a pretty big deal when it comes to supplementations as a pre or even a post or during training type of supplement. They've shown some promise, there's no doubt. We'll talk about that later in the term as well. So from this, just be mindful that there's a lot of amino acids involved in this process, and there's surely more. This is just sort of a close-up of the endoglycolysis and the Krebs cycle. I threw this in and probably in the wrong place, but I just wanted to emphasize this whole process of gluconeogenesis. And the definition is a process of synthesis of glucose or glycogen from non-carbohydrate sources. So it's not always protein that it comes from. And so substrates for gluconeogenesis, there's several. Amino acids, yes. Glycerol, we talked about glycerol, right? And that's sort of half of a glucose, but it still counts. Pyruvate. Pyruvate can go backwards in the process of glycolysis, as can lactate. Lactate oxidation, use of lactate as a fuel source is pretty significant. I think we've already talked about that in this class a little bit. Lactate can be converted back into glucose and be used for metabolism. It's a pretty big deal. So just be mindful. Yeah, gluconeogenesis isn't just coming from protein; it's coming from other products as well. All right, turning a page a little bit. So we're stepping away from the nuts and bolts of metabolism, and we've talked now about carbs, fats, and proteins. Hopefully you have a better idea of how they're being used and when, maybe not so much when, but where. Let's talk a little bit about the control of these pathways, because it's interesting, right? I mentioned that at the onset, we have these allosteric enzymes. We have enzymes that can be turned on and off. Well, what's turning it on and off, right? What are the signals to turn things on or off? If you could pause this video right now and take a second to think, right? What do you think would be a universal signal for metabolism to get turned on, right? Think about that, right? When we start to use our muscles, right? That's a sign that we need to start exercising, or that we're going to be exercising. What is universally created the more we use our muscles? Pause it for a sec. All right, now that you're back, right? Hopefully you said ADP. It's right there in the middle of the screen. ADP is a stimulator of every metabolic pathway. Why is that? Because when we start to use ATP, the automatic byproduct is ADP, right? Yes, we're going to end up with inorganic phosphate, which does stimulate some systems as well. And in extreme cases, we may end up with AMP, right? We talked about that a little bit with the phosphagen system. That will also stimulate metabolism. But ADP is that universal signal. It's like, oh yeah, if there's ADP around, that means we need to regenerate ATP, and that's going to turn on metabolism. If the purpose of metabolism ultimately is to just regenerate ATP so we get to that homeostatic place, then ADP must be that universal stimulator, and it is. As you look across this as well, let's look on the other side. Well, if ADP is a universal stimulator, what would be the universal inhibitor? Pause the video, think about it. All right, now that you're back, I know you didn't pause it. What would inhibit it? ATP. If ATP concentrations are at a homeostatic place as ADP would be, right, when we're at rest, there's no reason to turn on metabolism, right? So turning on those rate limiting enzymes, if we had adequate ATP, would be disastrous, right? If we had ATP concentrations at their maximum and PFK was continuously turned on and we kept burning glucose, guess what? We'd run out pretty darn quick. And that would be catastrophic to us as humans, because we ultimately probably wouldn't be able to eat enough to stay alive. Metabolism would burn through our energy that quickly. So again, this gets back to the importance of being able to turn on and off these rate-limiting enzymes, and being mindful of the things that do it. And so the inhibitors on the inhibitor side, right, ATP kind of makes sense. If we have adequate creatine phosphate, we don't need glycolysis as much because we know we can use a phosphagen system, so that actually slows down PFK. What else? A drop in pH does actually turn off PFK, and that's a little bit problematic. For those of you that follow along in your exercise physiology classes, a drop in pH is sort of a byproduct of using glycolysis, right? Wait a sec. So you're saying if I use glycolysis, pH drops, which turns glycolysis off through PFK? Yeah, that's kind of what's going on. It's one of those great questions I'll ask you maybe in this class, more next phys. Why can't we sprint forever? Well, our engine that we use when we sprint, which is ultimately glycolysis, turns itself off. Once pH starts to drop too much, whether it's the brain that slows us down or it's actually happening in the muscle, that's a great debate to have. But either way, that drop in pH does lead to fatigue. Part of it's because PFK gets turned off. So anyway, so just be mindful of those metabolic pathways. They're the metabolic pathways inhibitors and stimulators. This graphic kind of does the same thing at the next level. I won't pick on you on the information on this slide. I think it's a really cool slide though, and I do love to check it out. And it sort of peels back another layer and go, okay, there has to be more stimulators and inhibitors than were on that previous slide. Yeah, there are. They throw some hormones in here. You'll notice epinephrine being up here, cyclic AMP, glucagon, or so we throw in, and maybe that's the biggest difference between the two. Throws in a lot more of the hormones, and it kind of pins up exactly where in metabolism they play a role. So pretty cool slide with the activators and inhibitors on it as well. So we've mentioned hormones a bit, and they do play an important role in the regulation of metabolism, without a doubt. Things like insulin, things like glucagon, those have to do with energy release and energy storage for the most part. Your epinephrine and norepinephrine and cortisol, they have to do with the release of energy and even the stimulation or the activation of pathways to start metabolism or to keep metabolism going. So those are kind of your stress hormones, which when we're exercising can play an important role in keeping us going. Um, yeah, growth hormone will stimulate lipolysis, which a lot of people use exogenous growth hormone for that reason. It'll also stimulate growth, and stimulate anabolism, so growth of potentially muscle tissues. So I just put this on here again. This, like the previous slide, I'm not going to pick on you on these too much. They're there and they're definitely an important part of the picture, but I don't want to lose sight of the purpose of this class. And if I start to get into every awesome hormone, we're just never going to make it out of the metabolism talk. So just be mindful of those. All right. So this is stuff that we should have an understanding of, because as we move forward in the class, the interaction of energy systems is pretty important. And so if we take a graphic like this, and it's very simplistic, but it has some value in its simplicity. And so we look on the y-axis, and it displays percent energy contribution. On the x-axis, we have time. And what the time axis that it doesn't really say is this is sort of all-out exercise. So if we do all-out exercise, so if we sprint our hardest and that sprint lasts half a minute, which is about as long as most people can sprint for before they fall over, what is the primary energy contribution from that? Well, it would be this yellow set of data pretty much, at least the first 15 seconds. The creatine phosphate system or the phosphagen system. Yeah, so short duration efforts of all-out intensity, definitely phosphagen system heavy. If it extends to, I guess in this case, probably past 15 seconds, and this is a fair representation, glycolysis is going to kick in, right? And if we put some of the pieces together that we've just talked about, yeah, about this far down, we're starting to run out of creatine phosphate. We're starting to probably see some AMP show up, turning on the PFK to start get glycolysis going. Efforts lasting, half a minute to 2 minutes or so, all out efforts, right? We're talking a major contribution from glycolysis. So, for most of us, that's sprinting a lap on the track as fast as possible. For some of you, maybe that's 2 laps, somewhere in between. That type of effort, right, is going to be primarily glycolysis. You'll notice mitochondrial respiration or oxidative respiration, right? We're starting to use oxygen. Its contribution increases, and it's trying to contribute as much as possible. Really hard to get up to full steam, but when we start to see efforts that last probably more than 3-4 minutes before we fatigue, yeah, then we're starting to see a much more major role in terms of ATP regeneration coming from oxidative metabolism, the Krebs cycle, electron transport chain, probably the breakdown of fats coming in there depending on exercise intensity. So kind of a cool graph to just sort of, okay, how do these energy systems interact with each other? Is it just creatine phosphate and you're done? No, there's a transition. Just like with glycolysis, before glycolysis is completely fatigued, mitochondrial respiration kicks in. And something important to note, yeah, both of these sort of have a crossover before they're at least, down to a quarter tank. We're closer to, half of a tank, so to speak. So another graph kind of showing some similar types of things with these energy systems. If we look at the maximum rate of ATP generation, as you can expect, yeah, the phosphocreatine system gives us ATP like an explosion. The rate is incredibly high. Glycolysis, that rate is much lower. I would argue that the phosphagen system is probably more than twice as fast as glycolysis, but whatever. And then the oxidation of carbohydrates and fats. So this is sort of our Krebs cycle electron transport chain with 2 different substrates, either the carbs or the fats. Much slower, right? So if you're trying to run a fast race and you're only burning fat, you're not going to be getting ATP nearly fast enough to be running fast. Whereas if you're just relying on your phosphagen system, you got so much energy, you're going to explode off the start line, right? But what's the drawback? Well, as you'd imagine, maximum available energy. Yeah, the phosphocreatine system barely makes a showing on the y-axis there, right? As does glycolysis. Glycolysis doesn't necessarily give us that big of a bang for the buck in terms of how much energy is available before it limits itself. Total breakdown of carbohydrates, which does require that presence of oxygen, yeah, we got a fair amount. And that was sort of that, oh yeah, we can go 60 to 90 minutes kind of thing, sure. Fat breakdown, yeah, it is limited, but really, no one's going to get to that point. There's other things that are going to cause fatigue before we use up the available energy that we have in fat. So, just an interesting way to compare the two. Um, I think a final graph on sort of these energy systems and where our fuel sources are coming from. And I like this because it kind of helps us get a better understanding. Okay, when we're resting, where is our metabolism getting its energy from? When we start to exercise. And so exercise of varying durations, you can also consider this of varying intensities, right? 30-minute exercise duration is going to be higher intensity than 120 minutes. So if we look at that first, run, a 30-minute run, you're going to run as hard as you can. Some people can finish a 5K in 30 minutes. Man, that's a pretty good clip, right? About half of the energy that you are using is coming from glycogen from the muscles. A quarter is being split between plasma glucose and fat. As exercise duration increases, which means intensity is going to drop, we see an increasing reliance on fat as a fuel source, right? It goes to 36 to 45 to 62%, and a varying contribution of plasma glucose. Once we get past about that 90-minute mark, plasma glucose drops primarily because we're running out of it. So the body tries to continue to liberate it to make it available, but it's not always the case. That's it for our exercise metabolism talk. I'll see you next time.